So my next presentation is the development of alternative procedure in analytical sciences. Again, this is focused on the pharmaceutical industry. And again, the disclaimer that I have made earlier that my presentation are my views and should not be construed as views or policies of the USP, FDA, or ICH. In terms of the outline of my presentation, I will briefly discuss the validation, verification, and transfer of analytical procedure, which I will be presenting in the next presentation. Also, again, mention about the process of using a new analytical method or procedure. The one that I will be starting to describe to you in terms of the development of an alternative procedure for test procedures such as those in the identification, assay, and organic impurities. And finally, describe to you alternative procedures for residual solvents and elemental impurities. I chose this because these are the ones that are major in terms of the need in the pharmaceutical industry at the moment. So in terms of validation, verification, and transfer of analytical procedure, can we go back, Hazel, to the first one? Okay, thank you. So in terms of the validation, USP General Chapter again is validation is an established laboratory studies based on the performance characteristics of the procedure, and it is meant so that it will meet the requirements for the intended analytical application. 1226 is only selecting some of the analytical performance characteristics. It is not a repetition of the validation, but it is intended so that when you do verification of your product, it is trying to ensure that the lab is capable of performing the method reliably and precisely for its intended use. The transfer I mentioned to you earlier, I'm not going to repeat the transfer of analytical procedure again, but this is simply the from a transferring unit to a receiving unit, transferring an analytical method. It is there to ensure that the receiving unit has the procedural knowledge and the ability to perform the transferred analytical procedure as intended. And it is not applicable to compendial procedures or monographs. So I did describe to you the process of implementing new analytical procedure if it is a compendial. And this is shown by this slide. Similarly, implementation of an alternative method for non-compendial or alternative method, as I mentioned earlier, is shown here. And then the ways to deal with issues in terms of implementing can be using the system suitability, using developing the alternative or harmonized method and procedure, which is what I'm going to discuss in this presentation. And then the third one would be what is called the flexible monograph. So in the development of alternative procedure for procedures or methods for identification tests, assay, and organic impurities, you can use the first strategy, which is using the allowed variation in HPLC or the USP General Chapter 621 in chromatography. Or you can use the development by using robustness study approach, or you can use both A and B. But keep in mind that when you are developing an alternative procedure, it means that you have an initial knowledge of the procedure based on the monograph. So it's not, you don't have anything. You have something where your starting point would be the USP monograph, except you're having difficulty, so you're trying to modify the method. So in the development of alternative procedure, again, if you're using the General Chapter 621, remember that it is chromatography based. It is a system suitability adjustments. And the user should verify the suitability of the method under the new condition by assessing relevant analytical performance characteristics potentially affected by the change. So again, you can use these allowable adjustments without revalidation. However, if the adjustment is beyond the allowable range, then you need to do some level of revalidation. And finally, the advantage of this using the General Chapter 621 is that your adjustment may be a bit different from what is in the compendial, but it is going to, your alternative method will come up to be very close to what's in the compendial. So that eliminates a lot of your need to do method development, going back and doing a lot of the method validation. So in the system suitability adjustments, you have the list of variables listed here with the adjustment. So again, I mentioned to you earlier that you have to pay attention whether your system is isocratic or gradient, because some of them are only limited to an isocratic system. So I showed you this slide earlier, but this is more on the allowable adjustments, and then I'm highlighting for only those systems that are isocratic and isocratic or gradient. There is a difference between system suitability adjustment between different pharmacopoeias. If you're using the second strategy, which is developing alternative procedure for identification tests, assay, and organic impurities using robustness study approach. So robustness is actually a test procedure or a methodology where you examine the important parameters that affects your method. So for example, if you change the temperature, is it going to be critical? So those are what is termed robustness study. And the next table will show that sample preparation variables can be adjusted or can be revised based on the robustness studies. And these variables may be used as your starting point in the development of an alternative procedure. So in this next table, it'll show you the different factors, what you can do to adjust your procedure. Percentage of organic modifier, for example, you can, and also there's a suggested limit factor level. Sample matrix concentration, you can make this adjustment. pH of the extraction buffer, concentration of the extraction buffer, mixing equilibrium time. There are many of these factors that is listed in this table. Extraction temperature, dilution ratio, more type of mixing, shaking period, blending time, concentration of the active ingredients, filter pressure, discarding the filtration volume. So, and then finally, the equipment that you use. So you either use a mechanical shaker or blender. So these are all the different factors in terms of how you prepare your sample so that you can come up with an alternative procedure. And you, again, you can combine this with the first strategy, which is the system suitability adjustments. Now, how the next slides will be alternative procedure for developing residual solvents. So in USP, there's a general chapter 467 that describes to you the residual solvent analysis. Again, the De La Salle University Central Instrumentation Facility has this instrumentation called the Headspace GC. But in terms of the guidelines on the alternative procedure for residual solvents, it is consistent between ICH, European Pharmacopoeia, and USP that the manufacturer are free to select the most appropriate method to validate in terms of the analytical procedure analyzing for residual solvents. So meaning you do not have to analyze all the residual solvents, but you only need to analyze the residual solvents that are specific to your sample. So in terms of the overview of developing residual solvent procedure, you need to determine what solvents that you're going to be looking for, because there are a lot of solvents and there are 3 classifications. You need to choose the appropriate testing procedure for those specific solvents. Either you use what is prescribed in the USP General Chapter 467 as written, but you can modify them, or you can come up with your own method of residual solvent analysis. So when you're starting to do the residual solvents and you're the one who produces your own active ingredients or the API or the drug substance, ask the organic chemist what solvents they are using in terms of synthesizing the API. Ask the questions about what solvents they're currently using or what they foresee using in the future, so that you can prepare a methodology for residual solvents that will compile a list of solvents, all the ones that you will intend to look for. Now, if you are sourcing your materials from other vendor, you need to develop a supplier survey. Find out what are those residual solvents that the suppliers are using for the active pharmaceutical ingredients. Also, don't forget the excipients are important also if you're making the final product, because it would be good to find out from the supplier and request what is called the excipient information package. This is more like an MSDS for those excipients that you use to make your final product. Considering confirming the results in your laboratory, if they say it has, possibility chloroform, then you need to confirm that in your lab, and even develop a quality assurance, quality control procedure so as to determine that if an ongoing testing is necessary whenever you purchase this excipients. So every time you make a change in terms of where you source your material, it is a good thing to keep in mind. You need to check for the residual solvent impurities. An optimized solvent method is something that is accurate, sensitive, and fast, and you only have to analyze the solvents used in your process again. And you need to develop a methodology to meet your company needs. Now, this slide describes to you the 3 different classes of residual solvents. Class 1 would be solvents that are to be avoided because they have high toxicity. The second class are those that are solvents to be limited. You can use them, but they need to have some limited amount in the product. And then the class 3 would be the solvents that have low toxicity. So this slide describes to you the class 1 solvents to be avoided, and you see that the concentration limits are in the low ppm. The concerns are listed in this table, having the carcinogenic and toxic effect to humans. This next table may be difficult to read. Again, these are solvents that you can find in the book USP in the General Chapter 467. These are the class 2 with limited permissible daily exposure. And you can see also that the concentration limit is a bit higher. The third class, which is the solvent with low toxic potentials, are listed in this table. I just wanted to go briefly in terms of how do you analyze residual solvents, and the major instrumentation for residual solvents would be the gas chromatography, and to consider the 3 main components of the GC, which is the injector, detector, and the column. So make sure that when you're using the 467 described in USP, you can modify this, but it may require revalidation again for those conditions that you are changing in a major way. The injector can either be a headspace, which I think the Central Instrumentation Facility has this instrument, the headspace sampler. And this is the one that is described in the General Chapter 467. It has the advantage of reducing matrix interference, but the disadvantage is that some compounds are not detected using this methodology, headspace. You can do a direct injection in your GC, because it is sensitive and it detects all the GC and 467 solvents. But the disadvantage is it is subject to a more matrix contamination in the column. So injection can be split, non-split. So headspace is specified in the General Chapter 467 in USP because, again, like I mentioned, it reduces matrix interference. So most of the drug formulation products are not volatile. So it is a good instrument to use. The disadvantage is that some of the compounds are not detected. And some of these compounds that are not detected using headspace are listed here: formamide, 2-ethoxyethanol, the ethanol types, the glycol ones. And this chromatogram shows you why. Because the first two big peaks, which are the methylene chloride and the benzene, has partition coefficient values of less than 10. One is 5 and the other one is 3. The partition coefficient actually is a expression where the concentration of the residual solvent concentration in the solution phase divided by the concentration of the residual solvent in the gas phase. So notice that if the partition coefficient for the 1,4-dioxane is 1,618, you would see a very small signal when you do a GC analysis of it. So that would be the reason why a disadvantage of using a headspace analysis. You can optimize in terms of the partition coefficient. You can optimize the system by adding salts to minimize the variation in the partition coefficient. For example, you have different residual solvents in your sample, and this lists down some of the salts that you can use to minimize the variation. You can change the dilution solvent, for example. You can change the heating temperature, and also you can reduce the solvent volume so as to increase the concentration, because the idea here is you have a sample dissolved in a liquid and you're heating up the sample. It takes time for it to equilibrate, but the smaller volume you have, the more concentration you have, but the molar ratio would be the same in the headspace. So the headspace is the one that you actually inject in the GC. In terms of equilibration time, you need to determine the equilibration time of your sample experimentally. So it is the ideal time is determined experimentally, but most of the solvents are ready after half an hour. So you would create this determination in your laboratory where you determine the response, monitoring the time. So again, it levels off after some time, and that is what is termed as the equilibration time. The General Chapter 467 as written can be optimized, again, so that a general protocol is intended to meet the needs of everyone. It has long analysis time, so you can modify it to shorten it. The sensitivity requirements are hard to meet for certain compounds. So again, it allows you to develop an alternative procedure. And at times, like I mentioned earlier, it does not detect all class 2 solvents because of the partition coefficient issue. It is not capable, it's not applicable in terms of some of the drug formulation. In terms of the GC detector that you are using, the ones that are typically used for residual solvents as described in the General Chapter 467 would be the FID, flame ionization detector. Shown here, it has a wider range, but a good one to use would be a mass selective detector. And I believe Central Instrumentation Facility have this detector also, the mass selective detector. So a mass selective detector for GC has several advantages. It has high specificity, and it is good in terms of whether you are using the scan mode. It provides you at low ppm and even parts per billion in the SIM mode. And also the peaks are identified because of their masses. So it helps you in the identification, helps identify other unknown impurity peaks in your sample. So these are the advantages of having a mass selective detector. But you would need an internal standard so that you will have a high precision analysis. The disadvantages are several. It's an expensive instrument. It requires maintenance, so high maintenance costs. Also the training in terms of who's using this instrument requires time and some effort. Also, it's difficult to match the accuracy and precision obtained from other detectors such as the FID unless you use an internal standard. In terms of the column that you are using for alternative procedure for residual solvent, consider in terms of the inside diameter of your GC column. If you are trying to have a high resolution, you can use the 100 micrometer inside diameter GC column. It will provide you with good resolution. However, in terms of capacity, it's not going to be as much capacity, meaning if you have a lot of the residual solvents, the one that will provide you with better capacity in terms of the number of residual solvents you're using would be a wider diameter called GC column, so something like 530 micrometer. So it has good capacity, but in terms of resolution, because the wider the diameter of the GC column, the lower the plate number. So the resolution is not as good as the smaller diameter GC column. In terms of the length of the column, as a starting point, the suggestion is to use something like a medium length GC column, somewhere in the 30 meters, because you can use a long GC column providing you with high resolution, but it's going to take you a while because it's going to be a long analysis. The opposite would be using a short column that has moderate resolution, but it's faster. So this would be the recommendation in terms of developing alternative procedure for residual solvents. So optimize your GC method so as you can save amount of time, and that many changes could be considered as an adjustment to the 467 general chapter, but some may require revalidation, and revalidation time is significantly reduced if your method runtime is also reduced. The benefit of an optimized method may outweigh the extra validation work. So now we go to the last alternative procedure development that is also gaining a lot of attention in many of the pharmaceutical industry, and this is the elemental impurities. Now, the old chapter for elemental impurities described in USP is a chemical reaction, and this has been replaced by two instrumentation. One would be, the procedure one would be the ICP OES, optical emission spectroscopy, and then the other one would be the ICP mass spec. And I believe, again, the Central Instrumentation Facility have this ICP-MS as an instrument for elemental impurities procedure. This provides these two methods that are instrumental, provides greater clarity, especially for those who are trying to develop an alternative procedure. So actually the 233 general chapter actually has an actual procedure for elemental impurities, either for ICP OES or ICP mass spec. But if the specified compendial procedure listed in general chapter 233 do not meet the needs of your specific application, you can develop an alternative procedure. The alternative procedure, however, must be validated and make sure that it is so suitable for your own use. So alternative procedure either can be limit procedure. So limit procedures are those that gives you yes or no results. And the quantitative procedures are the ones that gives you an actual number in terms of concentration. But there are certain requirements even for limit procedures. So for limit procedures versus the quantitative procedure, limit procedure must be demonstrated experimentally using an appropriate system suitability and a reference material. So when you're developing a limit procedure, you know exactly what are the concentration, even if the final approach is a yes or no answer. So there must be known concentration to the yes, and there must be an appropriate acceptance limit of concentration. Also, the material or the mixture under test must be spiked. So spiking allows you to be specific in terms of your elemental impurities. For quantitative procedure, it would be the same, but the limit procedure must be done with repeatability, 6 independent samples, and acceptance criteria RSD is not more than 20%. In terms of specificity for limit procedure must be able to unequivocally assess each target element. So there's specificity even for limit procedure. For quantitative procedure, however, you need to establish accuracy. And accuracy would be a spike and recovery experiment. Somewhere between 70 to 150 would be acceptable for mean of 3 replicate preparation for each of the concentration. In terms of precision, when you're developing a quantitative procedure for elemental impurities, the repeatability can either be using 6 independent samples of material under test, and then the acceptable criteria is an RSD of not more than 20%. You need to do intermediate precision also, which is a different day, different instrumentation, different analysts, and an N or a number of analysis for 12 would require an RSD of not more than 25% for each of the target element. So quantitative procedure also requires specificity. And this specificity is produced, must be able to unequivocally assess each target element in the presence of other components. So you need specificity. In terms of limit of quantitation, range, and linearity, it must be demonstrated by meeting the accuracy requirements for the quantitative procedure. That's all I have for the development of alternative procedures, starting with the identification test, assay, organic impurities, residual solvents, and elemental impurities. Thank you for your attention.